So what I'll tell you about today are their efforts um, towards new treatments for bleeding and for thrombosis. So all related to blood coagulation and the lipids and RNA therapies we've been developing as part of the NMIN network. So as Christine mentioned, I'm an associate professor at the University of British Columbia, but actually our lab is moving to the Varsity Blood Research Institute at the Medical College of Wisconsin. And so most of all the work that you'll see today was done at UBC, and yeah, our lab will be at UBC for quite a bit longer during the transition. But we are moving to Wisconsin, and yeah, I do want to point out that yeah, we are recruiting postdoctoral fellows, graduate students, and technicians to Milwaukee. A little bit of change in scenery from Vancouver, but it's still beautiful here as well. So the big question that the lab's been working to answer, and the NMIN that we're doing, NMIN fits within this, is how do we control hemostasis in intractable hemorrhage? So the worst cases of hemorrhage. So hemorrhage is actually trauma, and hemorrhage is a leading killer of young people, and by young people I just mean below the age of 45, so maybe not not even so young. And hemorrhage bleeding is a big component of this. That happens during car accidents, during combat, during surgeries, during childbirth. Hemorrhage is a big issue. And so some of the questions that we ask are, what mechanisms fail during severe hemorrhage? And of course, can we address them? Come up with new therapies for controlling bleeding. In the schematics that I'm showing here, I'm showing two situations. It's the same size injury, and it's a big injury. So this might be a puncture to the femoral artery, so a very severe injury. But in the schematics, there's on one side it was manageable, and on one side it wasn't. It was intractable. So good things happened here in the manageable side. So in this large injury, several good things happened. So an external hemostatic was expertly packed into the wound. So a gauze material with a compound on it that was hemostatic was actually pushed into the wound, and it did its job. It helped form the blood clot, helped activate platelets, helped seal the injury. Platelets, they did their job in this case. So circulating platelets, they sensed they sensed molecules in the wound, and they became activated and helped form the blood clot. And these components of the blood clot, like fibrin and enzymes that help form and cross-link fibrin into a self-assembled gel, they did their job. So they formed at the site of the injury and only at the site of the injury, and helped seal that wound. But these broke down in this intractable case. So the external hemostatic was flushed out of the wound. Platelets, they didn't do their job. They didn't become sufficiently activated. It could be because the pH of the blood dropped to 7 or below, or not enough activators of platelets like thrombin were generated. And fibrin and 13A, they didn't do their job either. So either they weren't formed during the coagulation cascade, the enzymes didn't like thrombin wasn't formed to generate fibrin, or they were degraded. So they were chewed up in a process called fibrinolysis. And so these bad things happened, and so this particular patient wouldn't have done very well, unfortunately. So of course, the big goal in our lab is to take cases of intractable hemorrhage and make them manageable. That's the overarching goal. Here's some examples of intractable hemorrhage, types of hemorrhage that can't currently be addressed. And so this includes abdominal hemorrhage, a non-compressible abdominal hemorrhage. And so if there's internal bleeding in the abdomen, this can be a serious problem, and because it can't be treated by just packing a material in, or can't be treated by compression that could be used for most other types of bleeds. And so this is a study that we did at the University of Toronto with the Canadian Department of National Defense and Lieutenant Colonel Andrew Beckett and Hugh Semple. And so this is a swine model of non-compressible abdominal hemorrhage, and it recapitulates what's seen in humans for this type of bleed. And so as you can see, the survival went from 100 and dropped really low. So most of the pigs here are dying, and they're dying within an hour. And so a lot of the approaches that we're developing for severe hemorrhage and that have implications for bleeding disorders and thrombosis, we're trying to, to control clotting, we're not necessarily trying to make it 100%. We're not trying to make an intervention that you're in a serious accident and you don't need to go to the hospital at all. That's not necessarily the goal over the next 5-10 years. But if we can just shift this, curve and extend the survival time between less than an hour to be up to 2 hours, this will be really beneficial and give time for the person to get from that accident to a surgeon. So for this type of injury, it can only really be stopped by a surgeon. And so increasing the survival time is the big goal. A second big goal happens in the days or weeks after this severe bleed. And so what happens is that there's dysregulation of blood clotting. And so blood clots end up forming where they're not supposed to. This is called thrombosis. And so a second big goal is decreasing thrombosis that happens days or weeks after the injury. And there's many other situations where thrombosis occurs. And I'll mention one of them in a moment. So these are problems. Severe bleeding and thrombosis. These have been problems for a long time, and there's been many approaches, but it's still hard to still can't address these effectively. And so we really need new tools to modulate coagulation proteins and those related to coagulation, like the ones that degrade clots, the fibrinolysis. And so we need new tools. There's several areas where that we work in my lab, and of course today I'll talk about these two approaches. But here's the areas that we work. So we investigate biochemical mechanisms in hemorrhage control, trying to understand what leads to severe bleeds and less severe bleeds, and the biochemistry, the enzymology there. We create devices like powders and sprays for hemorrhage. And we also look at biochemical mechanisms in clot degradation, where clots are chewed up. Related to NMIN, we develop RNA therapies to try to modulate the coagulation system. And so we make RNA therapies based on those that are similar to those that are clinically approved, that go to the liver and either decrease the concentration of proteins in the liver or increase the concentration of proteins or make new proteins. And so we do this for thrombosis, and we also do it for bleeding disorders. And we're also using RNA therapies that I'll talk more about in a moment for modifying platelets. So bags of platelets are normally transfused into people, and so we're working to modify those bags of platelets. All towards this question, how do we control hemostasis, blood clotting in intractable hemorrhage? And so I'll start by telling you about the work that we're doing for thrombotic and bleeding disorders. And so these are the members in my lab that have done the majority of the work for this portion of the talk. And so this is Li-Jin Zhuang, Amy Wong-Strilchek, and Jerry Leung. And outside my lab, there have been many contributors. I wanted to highlight one in particular. So Peter Cullis, who's really a pioneer in lipids and lipid nanoparticles, as highlighted in a recent write-up in Nature. And so we collaborate closely with the Cullis lab. So one problem we're trying to address is mentioned is thrombosis. And there are agents to counteract thrombosis. These are called antithrombotics, but there's really a need for improved antithrombotics. And there's many examples of this. So one of them that's really been in the news lately is are the challenges of thrombosis highlighted by COVID-19. And so in COVID-19, if there's severe cases of it, this is often accompanied by thrombosis, and it's a major aspect of mortality in COVID-19 is thrombosis. And the antithrombotics that are used, even with the antithrombotics, there's still cases of thrombosis. So what's really important is that there's unfortunately there's major side effects of bleeding. So you're trying to administer antithrombotics to prevent someone from clotting, but then they have bleeding in about 5% of the cases. And this can be really serious bleeds, like in the brain, intracerebral hemorrhage. And so we've, over the years, we've been thinking about alternative approaches to thrombosis, and that's what I'll tell you about today. And so the question we asked is, can fibrinolysis, which is clot degradation, can it be gently enhanced? So instead of stopping the clots from forming, can we instead do a better job lysing them, cleaving them, and removing them gently. And can we do this using RNA knockdown of anti-fibrinolytic proteins? So we decreasing the concentration of proteins that prevent that degradation. And we use siRNA, so small interfering RNA, and lipid nanoparticles for this. This is an alternative strategy to what's currently done. And so you don't have to memorize this slide. These are the proteins and enzymes that help form blood clots. And what I'm just trying to, what we're really trying to show in this slide is that in the pathway from when the clot gets started in an injury by tissue factor all the way to the end where it gets cross-linked into fibrin, there's many steps and where these enzymes are being formed towards forming the clot. And the anticoagulants that are currently used, like heparin and direct oral anticoagulants, are also called DOACs, and ones in development, like an antisense oligonucleotide against factor 11. Each of these inhibitors is trying to prevent the clot from being formed. But an alternative strategy is preventing the stabilization of the clot. And so this enzyme down here, factor 13, it's a transglutaminase that cross-links the clot. And so we suspected that if we could inhibit that, we could destabilize clots that are formed to make them easier to cleave. Or we could directly decrease the concentration of fibrinogen, one of the main components of blood clots. Unfortunately, there are not good ways to target these antifibrinolytics, like factor 13, fibrinogen, antiplasmin, or thrombin activatable fibrinolysis inhibitor. So there's not great ways to inhibit these. And so we thought, well, maybe we could use RNA lipid nanoparticles to do this. First one, so there's two that I'll tell you about now, and then I'll switch and tell you about a third RNA therapy. So one of them is targeting fibrinogen. And so fibrinogen is a main component of main plasma component of blood clots. It's also an acute phase protein. And so it's increased during inflammation. In COVID-19, for example, our fibrinogen goes from a couple grams per liter. In severe cases, it can go to about 10 grams per liter. So it can increase fivefold or even more. So there's huge increases in concentration. This is an independent risk factor for thrombosis, and it makes clots resistant to being degraded. It also increases blood viscosity. And there are no effective long-term strategies to decrease it. It'd be really hard to make this is a protein and not an enzyme. It'd be really hard to make a small molecule to inhibit its activity and function. But I'll tell you about the strategy that we're taking to do this. And then another one I'll tell you about is enzyme I mentioned earlier, factor 13. So this cross-links clots to stabilize them. And there are currently no inhibitors for 13 that are suitable for in vivo use. And so these are challenging targets. And one point I really want to emphasize during this talk is that for proteins, especially proteins that are made in the liver, it's very straightforward by working with like the nanocore, an aspect of NMIN, to generate these nanomedicines that can inhibit the synthesis of these proteins. And so fibrinogen and one part of 13 are made in the liver. And so we formulated lipid nanoparticles that would deliver RNA to the liver. And so they're delivering small interfering RNA, which leads to the degradation of mRNA. mRNA is producing these proteins in hepatocytes in the liver. And so I won't go into the, unfortunately, not to disappoint too many of you, we won't go into the details of specific lipids that we use and the compositions, which you might be interested in. But instead, just say that, we use lipid formulations that were developed by Peter Cullis and Marco Cipollini at UBC. And there's similarities to those that are the formulations that have been approved by the FDA, such as in 2018, the Onpattro formulation. And so, this is just an example. So we use ionizable lipids. And so these are under acidic conditions, they are positively charged, and they'll complex with negatively charged RNA. And then when you bring them up to a physiological pH, the surface becomes neutral. And so that we don't use this particular lipid in it, but this particular lipid is ALC0315. It's in the Cutis BioNTech Pfizer vaccine, and so here's the ionizable group here. Okay, so we formulated these RNA into lipid nanoparticles, and then injected them into mice and into other animals. And that's what I'll tell you about soon. And so we had to do some tricks to come up with a siRNA against factor 13. And so I won't tell you about that, but happy to discuss this with you if you would like to target proteins made in the liver. But here's how effective it was, even just on the first try after we designed the siRNA. And so this is looking at the amount of mRNA in the liver of these mice after an IV injection at certain time points. And right away, the amount of mRNA for factor 13 drops from 100% to 10%, and after a few days, it's even below 10%. So right away, we can remove a lot of the mRNA from the liver. And if you look at the amount of protein that's circulating in the blood, that's also decreased. And so within a couple of weeks, it's brought down to 10% or below. And by injecting every 3 weeks, similar to the approved Onpattro therapy, we can keep the concentration of this protein low indefinitely. It's similar with fibrinogen. The fibrinogen we can bring, depending on the siRNA sequence, we could bring it down to various levels, including one that takes it to 15%, or with certain ways of formulating it, can even bring it down to 5%. And so this is looking at the amount of mRNA in the liver, and if we look at the amount of protein, circulating protein in the plasma, it's also the same. So the concentration has gone from 100 to 10% or below. And we can control this. So the idea is that if this is raised, if it goes high in inflammatory state, such as in sepsis or COVID-19, you can dial it back down to a normal level. And this has an influence on the blood clots. And so in this slide here, I'm showing a coagulation assay looking at the stiffness of blood clots. And so on this graph, 0 means that the clot has pretty much no stiffness, whereas 30 is a normal stiff blood clot. And so in gray is a wild type mouse that hasn't received any siRNA, and so the clot forms. It has this this stiffness, and then it degrades a bit over time. But the siRNA against fibrinogen, most of the fibrinogen in this case is wiped out, and so the clot doesn't, a complete clot doesn't form, and it degrades very quickly. And then with the siRNA against factor 13, we can still make a stiff clot, but then it degrades fast. And so depending on the situation, you might choose one over the other. So we expect this one to have a much less bleeding risk, for example. Okay, and this data just quantifying that the clot stiffness and how fast it's lysed or degraded. And so we looked at mice did a bleeding assay where there's a tail transection that's done to look at the amount of blood loss to see if these lead to excess bleeding. And so we were surprised actually when we decreased the amount of fibrinogen and looked at the amount of blood loss, that the siRNA against fibrinogen, if we look at the blood loss there compared to mouse directed with just PBS buffer as a control, there wasn't a major increase in the amount of blood loss. So we were surprised to see this. So this means that there's definitely compensation here. The platelets are being really effective and doing their job to stop the injury, even if there's not much fibrinogen. And when we looked at the siRNA for 13, and we also saw that this didn't lead to much excess blood loss. And so if we look at untreated mice versus those treated with siRNA for 13B, there's similar amounts of blood loss. But if we look at clinical anticoagulant, like this direct oral anticoagulant, apixaban, that one has much more blood loss. So we're encouraged to see that. There is interesting biology that's occurring here, though. That in the untreated versus the siRNA treated mice, even though the total blood loss is the same, it's interesting that there are a lot of rebleeds. So the clot would form and break and form and break. It would keep going on like that for long periods of time, and so there's a lot of rebleeding that was occurring. but the total amount of blood loss was the same. So we've done a number of in vivo studies now looking at the efficacy of these agents in various conditions. I'll just show a couple of those. I'll show one with siRNA against fibrinogen. And so with this one, we're looking at models of inflammation. And so we inject the mice with the siRNA fibrinogen, 1 week prior to the aspect of inflammation. And so in this one, we injected siRNA fibrinogen 1 week prior to injecting the mice with lipopolysaccharide, so LPS, to induce sepsis. And what we saw is that, as expected, the amount of fibrinogen went up during this inflammatory challenge. So it went from around 2 grams per liter to a bit over 3 grams per liter. And this is a problem during these inflammatory states. If fibrinogen increases, this can lead to thrombosis. What we found is that the mice on the siRNA fibrinogen had much less fibrinogen. So it decreased it back into what could be considered a normal range. So it had 1 gram per liter of fibrinogen. Now, okay, you may notice that we did inject the siRNA before this inflammatory challenge, and so in this case, this might not exactly mimic something that would necessarily clinically relevant in this case. You wouldn't know that you're going to get this infection and get an injection beforehand. But we do think there are applications for it in states where there is a chronic increases in fibrinogen. Excuse me. And this other model of a fibrinogen-mediated inflammation, this one's a bit interesting. Here we inject a chemical, thioglycolate, into the peritoneum and induces an inflammatory state. And during that inflammatory state, monocytes and macrophages, they home to that area to try to heal that damage. But there's a mouse model where it's missing a protein, plasminogen, and there the cells don't migrate anymore. What happens with siRNA fibrinogen is that it relieves this issue and allows the cells to migrate again. So these are experiments that are led by collaborators addressing biological questions they have, but it just shows that these siRNA lipid nanoparticles are effective in the hands of collaborators at knocking down the proteins and influencing these models of inflammation. I told you we were interested in thrombosis, and so I want to show you data looking at thrombosis in mice. And so this is with factor 13 knockdown. And so the first thing that we did is we wanted to understand what does this do to the blood clot. So first I'll show you that. And so in these plots here, we're looking at a protein that's in their blood called antiplasmin. And we're looking to see if this protein gets cross-linked to the fibrin clot. If it does, this will stabilize it from degradation. And so in the untreated mice, if we look at this Western blot for antiplasmin, what we see is that the antiplasmin originally has a certain molecular weight, but then if during blood clotting gets cross-linked, it gets to a higher molecular weight, it gets cross-linked to fibrin in the untreated mice. And the siRNA treated mice, so it doesn't happen. So without factor 13 to cross-link them, we don't have these high molecular weight species. And so this is the mechanism by which this particular agent is working. It's preventing this antifibrinolytic from getting cross-linked to the fibrin clot. And this has an influence on thrombosis in these mice. And so in this model, we made clots in the carotid artery of the mice and looked at the amount of blood flow through there. And so on the y-axis of these graphs are looking at the amount of blood flow. And when we make and so there's a high blood flow. When we make the clots with ferric chloride, the amount of blood flow drops. So there's very much less blood going through these carotid arteries. And so, but if we add an agent to help a clot busting agent, a very, very tiny amount, if the mice are on the siRNA fibrinogen, the clot opens back up. The clot is busted, it's lysed, and blood can go back through. But with the untreated mice, that doesn't happen. They stay closed. and that's what's quantified here. So with siRNA luciferase, which is a control, we don't. So in this siRNA luciferase control, the blood vessels stay occluded. There's thrombosis there, and it's a problem. It would cause could cause permanent damage and a stroke. Both the siRNA for factor 13, in most cases, this thrombus is able to be lysed and degraded and to allow blood back through the carotid artery to the brain. And so we're encouraged by this. And then the next step for this project, we're assessing this agent in the rabbit model of extracorporeal membrane oxygenation, or ECMO. And so in ECMO, if a person, in many cases these are pediatric patients, if there's a problem with the heart, the lungs, and you need help oxygenating the blood, take that blood out of the patient and circulate it into a device where the blood gets oxygenated and removes CO2, adds oxygen, and goes back into the patient. And this is a great therapy, but really susceptible to thrombosis. The device gets clogged, and this can lead to clots breaking off and really causing really major problems in the patient. And so at the moment, we're doing a rabbit model of ECMO with collaborators in Cincinnati. And so yeah, I hope to have results from this soon. We did, of course, first developed an siRNA that worked in the rabbits. And so we had one that worked in mice. And so we changed that sequence for rabbits. And we were really encouraged that we could also decrease the amount of plasma 13. And this decreased the amount of plasma 13, and this lasted for many weeks actually. And it still works by inhibiting the cross-linking of this antifibrinolytic antiplasmin to fibrin. So again, it's keep making clots. Clots still form, but they lyse much easier. Okay, and so we've been developing a growing library of RNA that are available to collaborators, and it's mostly collaborators in areas of coagulation, so thrombosis, bleeding disorders, hemorrhage, and inflammation related to thrombosis. And so we've made a number of agents for controlling them. I won't discuss them today, but usually ask collaborate, people in the blood field, if there's any of these that you like or any others you'd like, please let me know. We're very happy to share them. We're generating these because you really see major untapped opportunities for lipid nanoparticles in gene therapies for coagulopathies. And so on the top here, highlighting approved therapies that use LNPs, and down here, I'm highlighting gene therapies for coagulopathies like bleeding disorders and thrombosis. And so the approved therapies using LNPs, this includes Onpattro, which decreases the production of transthyretin a mutant form of transthyretin in the liver. It also includes, LNPs also include the ones made by Moderna and Pfizer-BioNTech Acutis for SARS-CoV-2, the vaccines against SARS-CoV-2. And so, yeah, there's lots of, of course, there's exciting things happening for LNPs and their development. And I think we can apply these to bleeding disorders and to thrombosis. And so here I'm highlighting the gene therapies that are in the works there. So there's clinical trials, both for hemophilia A and B, using adeno-associated viruses, so AAVs. There's also a phase II-B trial completed for thrombosis. This is an antisense oligonucleotide. And there's also a phase III trial for bleeding disorders using a siRNA, a GalNAc conjugated siRNA. And so, yes, I think there's, as I said already a moment ago, I think there's major untapped opportunities for lipid nanoparticle nanomedicine for bleeding disorders and thrombosis and for hemorrhage control. Of course, the advantages of RNA gene therapies are the rapid development, efficient manufacturing, and like point out, they can really also enable gene editing in vivo. And so wanted to, before switch over and tell you about gene therapies we're developing related to platelets, I wanted to tell you about one more target in the coagulation system that we've been going after. And that's plasminogen. And so you don't have to memorize this slide again, but plasminogen is the protein that when it gets activated by these clot busting agents, TPA, it becomes a clot busting agent and plasmin then degrades clots. And so plasminogen is a protein made in the liver that that contributes to the degradation of clots. And it's interestingly, inhibitors of this of plasmin are used extensively in the treatment of bleeding disorders, but they're not good. They have very short half-lives, and so it's one component of treating patients with bleeding disorders, but oftentimes a very small component of that because they're not good therapies out there. They have very short half-lives, and if you for patients that do take large quantities of these drugs regularly, a few times a day, there's a bad side effects associated with that because they have to take so much of this particular small molecule. And plasminogen deficiency is not a major risk factor for thrombosis, and so that's really encouraging. The adverse symptoms of patients that are missing plasminogen are reversible too, so that's also encouraging. As we develop these, we often look for that. So if we want to knock down a protein, we ask ourselves, okay, what patients are there already that have congenital deficiencies, and what phenotype do they have? And if it's a very if it's a really unfortunate phenotype, then we generally don't go after that target. Okay, and so to knock down plasminogen, we did make siRNA lipid nanoparticles and designed siRNA against plasminogen. And it was really encouraging what we saw. And so this is looking at the knockdown of plasminogen in mice. And so we look at the amount of concentration of the plasminogen protein. We can do the injection, just IV injection of the siRNA-LNPs, bring the concentration down below 10%. And through multiple injections, we can keep it below 10% for over a year, is what we've done so far. I'd also like to point out that we can extend this. So in addition to delivering siRNA, we can deliver guide RNA sequences against the plasminogen gene, as well as mRNA to make Cas9. And so this is work that's in progress and in collaboration with Drew Weissman. And of course, all this is in collaboration with Peter Cullis and the Cullis lab. But with this, so with this approach, you're delivering mRNA for Cas9 and doing therapeutic gene editing in the liver. And so we just started on this, but we can decrease the amount of plasminogen with this approach. And so one model that I'll tell you about is with mice that are a model of hemophilia A. So these have so patients that are missing or have very little factor VIII have hemophilia A. And so we took mice that are factor VIII knockout mice. And first we looked to see what the concentration of plasminogen would be with the siRNA plasminogen. And we found it was very effective. It could really even make it hard to detect plasminogen in this Western blot for plasminogen. And if we look at the amount of mRNA in the liver, it's below, well below 10%. And so we did took mice models of hemophilia A, and we did bleeding tests looking at the tail transections as well as injuries to their saphenous vein, and looked at the amount of blood loss. And what we saw is that the factor VIII, the hemophilia A, these factor VIII, they bleed a lot more. This is well known in the literature. So they bleed a lot more. But the siRNA for plasminogen can bring it back down to normal. So we were really encouraged by this, that it could correct this this bleeding phenotype in mice that had hemophilia A. I need to say, it didn't work in every model. Bleeding in mice is very different than bleeding in larger animals, and bleeds are over very fast and there's very little blood loss. And so we didn't see this in every mouse model, but we have seen, we always see trends, if you will, but in this case, it was very clear and statistically significant. And so where are we now with knocking down plasminogen? Right now we're doing this in a dog model of hemophilia A. This is the data from the wild type. So that's the in translating these to humans for therapies for hemophilia. Doing the dog models is a necessary step because the dogs mimic many aspects of bleeding disorders in humans. And so first we tested an siRNA against canine plasminogen in dogs, and we're after screening siRNA sequences, we were really happy to find one that really knocked down plasminogen, took all the plasminogen out of the plasma for over a month with one injection. And if we look at aspects of clotting, the stiffness of the clots, we find that by knocking down plasminogen, we can really make the clots very stiff, which is what you'd want if you're bleeding. You don't want soft clots that get degraded easily. You want really stiff clots, and this was able to achieve that looking at this clotting assay called thromboelastography. And so there's very little lysis occurring here, and we thought this could be really useful in dogs with hemophilia A, and those studies are ongoing right now. So we've just injected the first two dogs with hemophilia A. This is with collaborators, Tim Nichols at UNC and David Lillicrap at Queen's University. And David Lillicrap has become a member of NMIN as well. Okay, so that's what I wanted to tell you about on where we are with those with a few siRNA and gene editing LNPs for coagulation factors. And for the remaining 8 minutes, I'd like to tell you about work we're doing to modifying platelets. And this is being done by Jerry Leung. Stephanie Nowakowski contributed to our original papers on this work, and not in the picture, unfortunately, but hopefully you'll have a picture soon as Katie Bedior, Colton Strong, and Madeline Robertson contributed to the work that I'm going to tell you about in a moment. The question here is, can we express proteins in platelets, exogenous proteins in platelets? And people have been trying to do this for a long time, but it's tough because platelets don't have nuclei. A lot of methods that would normally be done to transfect cells and express proteins can't be done with platelets. They're also very finicky. They get activated very easily. So I'll tell you about why we want to do this and our successes so far in doing this. The collaborators for this work include Dana Devine, Eric Jan, and Peter Cullis at UBC. And so the question they're asking is, how do we, the big question, as I mentioned in the beginning, is how to control hemostasis and intractable hemorrhage. And platelets are a main component of this. And so if someone has severe hemorrhage, on transport to the hospital or at before surgery and during surgery, they receive transfusions of platelets that are very helpful, but many times are not enough. If the bleed is it's a really large, really severe bleed, the platelets aren't by themselves aren't enough. So can we enhance them? Can we engineer platelets, transfusible platelets, to express other proteins that can aid in their ability to stop bleeds? And so we had a few options for doing this. We considered injecting liposomes that contain proteins into platelets, or delivering LNPs with siRNA or mRNA to try to control the proteins in platelets. And what I'll tell you about today is packaging mRNA and LNPs, delivering them to platelets where they're endocytosed to then make proteins in the platelets, and the work that we've done so far in this direction. We think this is a very good approach for engineering platelets compared to alternatives. And so we're taking bags of platelets that people have donated, so donor platelets, and we think this is compatible with current blood banking processes at Canadian Blood Services and at blood banks such as Versity in Milwaukee. We also think this is easier to achieve than culturing platelets, or by doing bone marrow transplants to modify platelets. Obviously, you can't do a bone marrow transplant for severe hemorrhage. Okay, and so as mentioned, what we've been doing is taking LNPs with mRNA, loading them into platelets where they get endocytosed to then express the proteins. And we hypothesized that LNP mRNA optimizing LNPs and optimizing the mRNA sequences could lead to the expression of exogenous proteins. In the original work that we did towards this, what we found is that we could, if we removed platelets from, if we did certain procedures, we could load platelets with the liposomes. They would get endocytosed. We could also load them with mRNA. And so, but at that time, they actually didn't express an mRNA. We could get it inside of the platelets, but we couldn't. An mRNA was not being utilized by the platelet. And at that time, we weren't sure. You know, is it a problem with being released from endosomes? Is it not a? Is mRNA not finding the ribosomes? Is there something wrong there? And so by optimizing the LNPs and by optimizing the mRNA sequences, we've been able to overcome this. I won't tell you the details of that part today. I'm happy to discuss with you, but I'll show you, just because I just have a limited amount of time, I will show you that we have been able to achieve this. So, so Jerry, Katie, Colton, and Madeline were able to do this. And so here's just showing mRNA for luciferase, as well as for GFP. And so with mRNAs loaded with luciferase, we can get expression of luciferase enzyme, and with mRNA for GFP loaded in the platelets, we can make parts of the platelets fluoresce in green from this GFP. And so this is a major project in the lab and ongoing, and happy to collaborate and discuss this one more with anyone if you'd like. Okay, and so the reason why we're doing this is to try to make platelets that are resilient and useful in hemorrhage control. And this is just a schematic that describes the experiments that we did in this paper, but early examples show that this is possible. And so this wasn't mRNA in the liposome. In this case, we actually loaded the enzyme thrombin in there into so liposomal thrombin that we load into platelets. And normal platelets, when they saw indicators of vessel damage, they clotted. So they helped form fibrin and became activated. And what we found with taking thrombin in liposomes, that we had, what sometimes referred to as super platelets. So they generated clots very fast. They made really stable clots that weren't degraded easily, and they contracted really well. The platelets contracted and made a very stiff clot. And so really encouraged by by delivering the enzyme directly, and we think that expressing the protein and enzyme is an even more powerful approach to modifying platelets. And that's what I'm doing next. And so I'll end here, and we'll be really happy to take questions. Of course, if you're interested in going stateside for a little while, very happy to host you. And yeah, I'd like to thank this lab members again, and of course our collaborators and the funding sources, and especially NMIN really helped get these projects off the ground. So yeah, thank you. Yeah, thank you, Christine, and yeah, thank you everyone for attending.
Thank you so much, Christian. What a great lecture. Really appreciate it. And I've tried to block out that you're actually leaving us. So it's difficult to see you go. You've played such a huge role. Well, I mean, it's a big loss for us in our community in Canada broadly, more broadly than NMIN actually. So, but I do understand you're still going to stay involved in the network, right?
I hope so. Yeah,
okay,
as long as you'll have me.
Absolutely. Delighted to have you. So we do have one question already in the Q&A section here, and I invite others to share questions. So one here is by Jen Yi has put forward, and they're asking, what is the target organ for your LNP RNA delivery, and is there any target ligand design on the formulation? So I think looking to see if there is a target, an active targeting strategy that's being used.
Great. Yeah, thank you for that. On the LNPs that we use, we haven't attached any targeting moieties to them, and yeah, I've dabbled in this area a lot over the years. Personally, I haven't been able to have a lot of success in that. So hopefully in the future will be. So we rely. So these systems, which pioneered by others in the NMIN network. They rely on natural mechanisms. So when the LNPs, when they go into the bloodstream, they bind ApoE, and that helps direct them to be endocytosed by hepatocytes. So here it's all in the liver.
Liver,
Of
right.
course, there's a huge, exciting opportunities in other organs, and we're really interested in trying everything we can to contribute to that. But luckily for me, working in blood clotting, most of the proteins in blood clotting are made in the liver. So I'm pretty content with the liver at the moment in some ways. But
Great, thank you. Does anyone else have any questions? Please let us know. What I love to see here is the important role that nanoparticles, nanomedicines, formulation science is playing in your work. Can you talk about that a little bit?
Sure. Yeah. Over the years, I always dabbled with nanoparticles, but our focus, really is in blood coagulation, right? And so always looking for new strategies in order to be able to control clotting. And yeah, it's amazing how well these nanomedicines work for doing that. Every time we create an LNP, with RNA in it, always surprised how well it really does, like, knock down the target or make the protein in the liver or get delivered to cells to do it. So yeah, the role of nanomedicine and helping us achieve our goals and thrombosis and hemostasis are these are amazing technologies.
Great. So to what extent do you think, do you see some of this work being translated and commercialized? How far off would that be?
Yeah, that's these are good questions. For some of the other, so I like to contrast it a bit with some of the work we're doing making devices for hemorrhage control. And so there are faster timelines, and these are external agents. So that's there's some so the commercialization we're doing in those areas is faster. For the LNP lipid nanoparticles, really the huge success of the COVID-19 vaccines, I think are really helpful in that. And so for us, we've been focusing on animal efficacy and trying to do that in large animal models, such as the canine model and in the rabbits. But yeah, there's still lots of other aspects to our preclinical translation. So safety testing, and then of course the GLP safety testing. So I think there's definitely talking about many years still. But it is a track we're on, and working with so Bayer was an early funder along with NMIN for this, and so trying to work with them, and then with Nanovation as well, of course, trying to move these forward.
That's great, thank you. So now I can see all of the questions are coming in. We have Shubash and Thea that's asking a question, saying, great talk. Absolutely, it was. Just wondering about the expression kinetics of mRNA and platelets. Is it similar to normal cells? Can you please comment on this?
Yeah, thanks. It is similar. So we can, within at least it's similar to what I've seen in hepatocytes. So yeah, if we deliver the mRNA within a few hours, we can detect the protein, and about, I'd say, around 10% of the total protein the maximum amount of protein we're going to get. And then in 24 hours is where we're seeing the maximum amount, and then it'll wane from there over a few days or so. So which is similar to hepatocytes, and so in vivo you can start to see the same thing. You can start to see the protein getting made within hours, and then the LNPs, they might slow things down a bit in vivo, so you can see that expression for days before it starts to wane. And so it's similar in platelets as what we see in hepatocytes, for example.
Okay, great, thank you. And another question, would it be possible to extract the extracellular vesicles of the engineered platelets instead of gene-loaded LNPs for better targeted gene delivery? Do you think that would make a difference?
Yeah, it's an awesome thought, and it's something we've considered, but other people are. I don't think anyone's tried exactly that yet. but it is an area where people are interested. So there's a for example, there's a company in Boston that's taking extracellular vesicles from platelets and trying to use them for various applications. But loading those extracellular vesicles and using them as a drug delivery tool, I think is a great idea that considered but haven't pursued heavily yet.
Okay, great. Well, that was Morteza Kafshgari's idea. So just to let everyone know. So fantastic. Okay. Maybe a new grant application is emerging from listening to your talk today. And another person is asking, how do you overcome the opsonization of these LNPs? Is that something that you're thinking about?
Yeah, no, it's not something not something I thought of. Yeah, I'm not sure. I think that's a good question. I don't have an answer for that. But yeah, I mean, in all cases, the LNPs are, even in platelets, they still get coded. Yeah. That's a good question. I don't have a good answer to that.
Well, that's okay. Good. And one last question before we wrap up for today. Could pH-sensitive molecules help in controlled release? Or do you plan to have bulk release? That's the question here.
Yeah. Yeah. So definitely the once the LNP, I think part of the assumed mechanism for these is that when they are endocytosed, they're in endosomes, and then if the pH drops in lysosomes, that drop can help protonate the ionizable lipids to help with release from the endosome or lysosomes. But I'm not sure if that answers the question. I'm not sure you're asking if something a bit different than that, but that's assumed to be part of the mechanism of delivery into cells. But yeah.
Perfect.
I'm not sure beyond that.
No, that's great. Thank you very much. I think there's a lot of questions related to the formulation, as you might imagine. Well, that's perfect. Thank you so much, Christian. This has been such an interesting talk. We appreciated your time today, and thank you everyone for attending.